Hello students, myself Chinalisha welcomes you on behalf of Saraswat International Academy. Today we will begin with the last part of the chapter, Principles of Inheritance and Variation. Students, in the last lecture we have studied regarding the various types of the mutations. Today we will begin with the topic pedigree analysis. So basically, in a simple layman term, what is a pedigree? Basically it is a family tree. Now here, what is the purpose of this family tree? So pedigree, it is a family tree which is drawn with standard genetic symbols showing the inheritance pathway of the specific phenotypic characters. So basically a pedigree is a family tree which is having certain genetic symbols. Which genetic symbols are used, we will see that. And with the help of that symbols, we can see the inheritance pattern of the certain phenotypic characters. So basically it is the study of the traits as they have appeared in a given family line for several past generations. How a particular character, how a particular trait, it have appeared in the family through several generations, that can be studied through the pedigree analysis. It is a kind of genetic analysis in which the trait is traced through several generations of family to determine how the trait is inherited. The propositus, it is the first identified case of an inherited disease in a family. Say for example, if a person is having a fused earlobe, right? So, who is the first person to show that particular fused earlobe? That first person is called as the propositus. Basically, when the patient is male, the word propositus is used, and if the patient is female, the word proposita is used. So basically, with the help of the pedigree analysis, the inheritance of a particular trait, it is represented in a family tree over the generations. In human genetics, pedigree study, it provides a strong tool which is utilized to trace the inheritance of the specific trait, abnormality or a disease. So basically, what is the usefulness of the pedigree analysis? We can study the inheritance pattern of a specific character, of a specific abnormality or of a specific disease. Now, I told you that in the construction of the pedigree analysis, that is basically a family tree, certain genetic symbols are used. So, which are the important symbols used in the pedigree analysis? We will study that. So, students, here you can see the basic figures. So, here you are able to see certain symbols. So, basically see the first symbol, it is a square. So, basically a square represents a male. Now, that square, it is not shaded, right? So, it means it is an unaffected male. It means that person is not showing a trait, a abnormality or any of the disease. Now, the circle which is unshaded, it represents the unaffected female. This particular rhombus like structure, what does it represent? So, basically it represents a sex unidentified, right, or unspecified. We don't know the sex of an individual. Now, if basically the individuals are affected, then they will be shown by the shaded figures. So, if it is a shaded square, it represents affected male. If it is a shaded circle, what does it represent? It represents the affected female. Now, a line between a square and basically the circle represents the mating. That is, there have occurred a copulation between that male and female. Then, a double line between the square and circle, it represents the consanguineous mating, which means the mating between the relatives. Now, students, I told you that the line between the male square and circle, it represents the mating. Now, there is a hanging line, right? And then again the square and circles are shown. So, basically these above are the parents and lower one are the offsprings. Now, students, the offsprings are mentioned in the order of the birth from left to right. So basically जिनका birth पहले हुआ होगा they will be placed on the left side and then accordingly they will be placed towards the right side. Now in this particular figure you can see that basically this square, it is the shaded. So basically parents are normal. See here, these are the parents, they are unshaded, but an child is suffering, a male child is affected with the disease. Now this number inside a rhombic like structure, it represents a number of the unaffected offspring. So basically they are, if it is 5, it indicates that there are 5 unaffected offsprings. See students, these are some other symbols. I told you this is unaffected male, unshaded figure circle means unaffected female. Now these two symbols, the circle and the square with the basically the slash through it, it indicates the deceased male and female, that is they are dead. Then a square with a arrow on its lower side, it represents the propositus. I told you what is propositus. It is the first individual to show that particular trait. The shaded square affected male, the shaded circle affected female. Now basically we will study what is X-linked disease. So basically those genes which are located on the X chromosomes, right? And the diseases due to that is called as X-linked condition. So basically a circle, we know what is a circle representing. A circle represents the female, right? Now here the circle is not completely shaded, it is shaded but basically not. So it indicates that the female is a carrier of particular disease. This particular symbol, I told you, the rhombus like structure, it represents the sex unspecified. This symbol represents the abortion or stillbirth, and the sex again is unspecified. A single line between the circle and the square, mating. Double line between the circle and square, consanguineous mating, which means mating between the relatives. Now students, always see basically the first generation, the generations are represented by Roman numerals. So see here, the 1 is written in Roman number and this again 2 is written in Roman number. So what does it indicates? That it indicates the generation. And the Arabic individual, the Arabic numbers, they represents the number of individuals, right? So basically here the above one are the parents, this second generation is of the offspring. So here the Arabic numbers are individual. In the second generation, the first one is the propositus, right? Now the two squares and in between them a line, it shows non-identical twins, and the two circles basically it shows basically the two circles, so basically they are female, and the line between them indicates that the identical twins. Now students, the genetic disorders. Till now we have studied pedigree analysis. So basically pedigree analysis is basically a formation of the family tree through with the help of the certain symbols through which we can study the inheritance pattern of certain abnormalities, of certain characters, or of certain disease. Now sir, today we will also study regarding the different kinds of the genetic disorders taking place in the living organisms. Now first of all, what is a genetic disorder? A genetic disorder, it is a disease or syndrome that is caused by an abnormality in an individual DNA. So if a disease occurs due to the abnormality in an DNA, then what is that disease called as? It is called as a genetic disease. Now, how such abnormalities can occur, or what are such abnormalities? So, students, these abnormalities can range from a small mutation in a single gene to the addition or subtraction of an entire chromosome or even a set of chromosomes. Now, students, we have studied regarding the mutations. So, through mutations, we can know how the abnormalities occur in the genes. There are two ways. Either there will be a mutation in a single gene, we have studied substitution, we have studied addition, deletion, right? or it may be either due to the addition or deletion of one chromosome, or there may be addition or the removal of a complete set of a chromosome. So, how abnormalities arise? Either due to the mutation or either due to the addition or subtraction of an entire chromosome or of an entire set of a chromosome. Now students, we know each and every feature in a living organism is controlled by one or the other gene located on the DNA present in the chromosome. Now we know that every character of a living organism, who controls every character? A gene. Where are these genes located? On the chromosome, basically on the DNA. And where is that DNA? Basically on the chromosome. So we know that basically DNA is a carrier of the genetic information. It stores the information of each and every character, and hence it is transmitted from one generation to another without any change or alteration. Normally the DNA, it transmits from one generation to another generation without any alteration. But yes, changes or alteration do take occasionally. There are chances that changes or alteration may take place. So, this alteration or change in the genetic material is referred to as mutation. We have already studied, in detail a complete topic of mutation. Number of disorders in human beings have been found to be associated with the inheritance of the changed or of altered genes or the chromosome. So, there are many diseases in human beings which occurs due to either changes in the genes or due to the changes in the chromosomes. And what are such disorders called as? Those all diseases or disorders which occurs due to the changes in gene or due to the change in the chromosome. All such disorders are called as what? Genetic disorders. Now students, the classification of genetic disorders. Now students, you can see here in this slide that there are basically two types of the genetic disorders. One is the Mendelian disorder and another one is the chromosomal disorders. So basically Mendelian disorders are the disorders which occurs due to the change in the gene, and chromosomal disorders are the disorders which occurs due to the changes in the number of the chromosomes. Basically the Mendelian disorders are due to the changes in the genes, and chromosomal disorders are due to the changes into the chromosome. So if the changes occur in the chromosome, they are chromosomal disorders, and if the changes occurs in the genes, then it is called as the Mendelian disorders. So first of all, we will study regarding the Mendelian disorders. So what are Mendelian disorders? Any alteration or mutation in a single gene. So if changes or alteration occur in a gene, then what are they cause? They cause the Mendelian disorders. Now basically the Mendelian disorders are transmitted to the offsprings on the same line as the Mendelian pattern of inheritance. We have studied the Mendelian pattern of inheritance. Parent generation, then from parent generation it is transmitted to the offspring. So the same pattern is followed by the Mendelian disorders. Now they can be traced. How can we trace this Mendelian disorder? So basically by the pedigree analysis. Now some examples are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. These are some of the Mendelian disorders. Now students, these all disorders may be either dominant, that is it will be transmitted from every generation to generation. They may be recessive, that is mutation basically in the recessive alleles, and autosomal, that is the affected genes may be located on the autosomes, or sex linked, that is the affected genes are located on the sex chromosomes. So basically we can classify this disorder into dominant trait, recessive trait, autosomal dominant, or they may be sex linked trait. We will see this. See students, in the next slide we will see what are those traits. So basically through pedigree analysis what we can study? So basically one can understand whether the trait in the question is dominant or recessive. It may be linked to the sex chromosome like in the case of hemophilia. So basically hemophilia is a sex linked disorder. How we will see that? And it is evident that this X-linked trait shows the transmission from carrier female to the male progeny. So as the genes are located on the X chromosome, normally what is the condition? Females will act as a carrier of disease. How we will see that? They won't have the disease, but they act as a carrier of a disease and may transmit the disease to the male offsprings. So now I told you category, the different types of the category of Mendelian disorder, or you can say these are the categories of the pedigree analysis. So which are those? So first is the autosomal dominant trait. So autosomal, that is what is affected? Autosomes. Now we know that autosomes are similar for all males and females. So each and every individual may be affected. Dominant, that is which gene is affected? It is the dominant gene. So basically if we study the inheritance or if we study the pedigree of this type of diseases, this pedigree it rarely or seldom skips the generation. As which gene is affected? Dominant gene. And as the dominant gene, if suppose a heterozygous condition is there, say for example a dominant gene and a recessive gene. Recessive one is normal, but we know that always which one is expressed? Dominant one in the heterozygous condition. So what is that called as? It is autosomal dominant trait, which will be rarely skipping a generation. Autosomal recessive trait. So basically it may skip the generation because suppose in a heterozygous condition we consider, say recessive characters are expressed when both the genes are recessive, right? When both the alleles, if they are recessive, then only the recessive character is expressed. But if along with the dominant and the recessive, that is in the heterozygous condition, what is always expressed? A dominant character is expressed, right? So if a mutation have occurred in the recessive gene and if the individual is heterozygous, then the person won't show the disease, that is it may skip the generation. Sex linked dominant trait. So basically it is more common in the females. Sex linked recessive trait, which is more common in the males. Now see students, why sex linked dominant trait is common in the females? We know that we have the two X chromosomes, right? Now if one say for example a particular disease is there and in that changes occur in the dominant gene. So of course though other is normal, but the changes have occurred in which gene? The dominant. So basically as we have two X chromosomes, this is more in common. Now X linked recessive, or you can say sex linked recessive trait. We have two X chromosomes and males have one X, basically females have two X chromosomes and the males have one X chromosome and one Y chromosome. Suppose the individual we are talking about sex linked recessive trait. Now suppose if we consider a female, so in a heterozygous condition, of course the dominant one will be normal and the recessive which is which is basically mutated and is responsible for causing the disease. But in the females they have two X chromosomes, right? So there is a normal X chromosome along with the X chromosome which is having some of the mutation. But who is dominant? The normal one is dominant. So females basically don't show the symptoms of the disease, they normally act as the carrier. But students, in the males they have only one X chromosome and the basically counterpart of that other is the Y chromosome. Now the genes present on the X chromosome and Y chromosomes are different. So if the X chromosome is affected and its counterpart is not an X chromosome but the Y chromosome, so males will of course show the disease. And Y linked trait, it means it directly passes from father to son. So genes are located on Y chromosomes and if of course single Y chromosome is there, so always they will pass from father to son. See students, this is a pedigree shown. The first one is of the autosomal dominant trait. So here you can see where are the basically the genes are located on autosome. So it is common that all the individuals may suffer. Now which gene is affected? Dominant gene. So see here, this is the first generation, this is the second generation, and these are the different 3 generations. This is the 4th generation. Now see, a mating between an affected individual and the non-affected individual. So here in the second generation you can see that we have the affected individuals. Now see, in the second generation we also have the unaffected individuals. Say for example, ये second generation का जो male है वो unaffected है, and that male is marrying a particular female which is again unaffected. So इनके बीच का जो generation है यहाँ पे कोई भी affected नहीं है. But see, the second generation, one of the female, basically the daughter, it is affected. That affected daughter marries a normal male. But see, I told you it is a dominant trait. So here it is not skipping the generation, right? But in the say for example, the autosomal recessive trait. Autosomal recessive traits are those traits which skips the generation. देखो, first generation में नहीं था, right? Second generation में you can see that they have the trait. यहाँ पे जो मैरिज हुआ unaffected individuals के बीच में, फिर भी उनका जो जनरेशन हुआ यहाँ पे आप देख रहे हैं कि one of the individual is affected. So recessive trait may skip the generation, dominant trait never skips the generation. Now we will study some of the disorders in detail. So first disorder is the color blindness. So what is color blindness? So it is a sex-linked recessive disorder. So basically first of all see the name, sex linked, that is its genes are located on the sex chromosome. Recessive character, basically they will be expressed when both the alleles are recessive. Suppose if one is recessive and another one is normal, then the person won't be affected. Now it is a sex-linked recessive disorder due to the defect in either red or green cone. Now students, you have studied that we are able to see the various shades of the color, and they are due to the presence of the cone cells in the eye. So basically the defect arises either in the red or green colored cone or cells of the eye. Suppose if the red color is affected, the person is unable to see the red color, right? And or basically it is unable to discriminate between the red and green color. Then if green is affected, then too is unable to differentiate between which is red and which is green. So resulting in the failure to discriminate between the red and green color. Now why this defect arises? It arises due to the mutation of the certain genes which are present on the X chromosome, and I told you it is which type of disorder? Sex linked recessive. So where are genes located? On the X chromosome, and the males have only one X chromosome, females have two X chromosome. So in a heterozygous condition, what will be the possibility? That the female will be which type of female? Unaffected. She will be the carrier but will be unaffected. She won't show the symptoms of the disease. Why? Because one of the chromosome is the normal one and another one is the affected, but normal is dominant over what? Affected one. It occurs in about 8% of the males and only 0.4% of the females. Why? Because the genes that leads to the red-green color blindness, they are located on the X chromosomes. Males have only one X chromosomes and females have two. So अगर male affected है तो उसका counterpart तो Y है, तो of course he will show the disease. But female अगर heterozygous condition है, so what is the condition? That she will be unaffected. She will be the carrier of the disease but won't show the symptoms of the disease. The son of the woman who carries the gene has 50% chance of being color blind. So basically suppose a person is a normal person, a male is a normal male, but a female is a carrier of a disease. Very very important. Next slide में मैं आपको ये inheritance pattern दिखाऊंगी. So female is a carrier of a disease, that is she is heterozygous, that is one of its X chromosome is normal and another one is with the mutated genes, and the male is a normal one. Now suppose इनके बीच में mating हुआ and there is a birth of a son. So there are 50% chance that a son may be a color blind son. How we will see in the inheritance pattern, I will show you. Now see, in this case mother is not herself color blind because the gene is recessive, and we know that when dominant and recessive is present, always dominant gets expressed. We are talking about Mendelian disorders, and in Mendelian disorders they follow the path of the Mendel's pattern of inheritance. So according to the Mendel's pattern of inheritance, what is the condition कि अगर dominant और recessive साथ में हो, that is heterozygous condition हो, तो कौन अपना character express करता है? Dominant वाला. So यहाँ पे when we talk about color blindness, normal chromosome is, or basically normal gene is dominant over the recessive gene. So कौन सा character express होगा? That the individual won't be the color blind if in the heterozygous condition in the case of the female. That means its effect is suppressed by her matching dominant normal gene. I told you normal gene is dominant over the recessive gene. So the daughter will normally be not be color blind unless her mother is carrier and the father is color blind. Daughter के color blind होने के chances कब है? जब mother carrier है and father color blind है, ये कंडीशन में डॉटर क्या हो सकती है? कलर ब्लाइंड हो सकती है. अदरवाइज देयर वोंट बी द पॉसिबिलिटी ऑफ डॉटर बीइंग कलर ब्लाइंड, राइट? See students, a woman is a carrier. I told you, I am showing you the inheritance pattern. Here the woman is a carrier and a male is a normal, and in that condition I told you that there are 50% chance that a son born through such child will be colorblind. Now see students how it is represented. So basically X C and X plus. So basically what is considered, the C is a gene for the color blindness and the plus sign indicates the normal gene in this condition, and male is normal, which means the X plus, that is he is having a normal gene with the Y chromosome. Now what are the gametes produced? Here XC and X plus, and here X plus and XY. Now see the possibilities of the generation, that is XC X plus. So basically a carrier daughter, X plus which is a normal daughter. Now see students, XC and Y, that is a color blind son, and this is a normal son, X plus and Y. So out of the son there are two possibilities, 50% may be color blind, 50% chances are that a normal son may be developed. Now students, I told you that if a carrier woman mates with a color blind man, then there are chances that a daughter developed through them may be color blind. Now see students, what are the eggs produced? What is the C is the condition for the, C is the gene representing the color blindness. So which type of the gametes are produced? The eggs are produced. See students, here there is one mistake, X, and here it will be XC. Listen very carefully. Two types of gametes, X and XC. And what are the sperms produced? So basically XC and Y, because the male is color blind. Now suppose this with this XXC, that is a carrier daughter. Then X and Y, it is a normal son. Then XCXC, which means a color blind daughter, and XCY, which means a color blind son. So if a carrier woman marries a color blind man, then there are chances that daughter developed through them may be color blind. Now students, the next is the hemophilia. Hemophilia, again the inheritance pattern is same as that of the color blindness. It is also a sex-linked recessive disorder where the genes are located on the X chromosome. So, normally female will act as a carrier of a disease and the males may have the disease. Now students, in this condition, the females normally won't have the disease. They will always act as the carrier of the disease. The reason is that suppose if both the genes are basically hemophilic genes in female, what condition arises? Normally ये जो genes होती है hemophilia की, if both the genes are recessive, then that condition is called as a lethal condition. Lethal genes है, lethal genes है, इसका मतलब ये होता है कि individual will die immediately. Lethal genes से there will be immediate basically death of an individual. So ये कंडीशन बहुत 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 रेयर होगी और होगी तो दैट फीमेल विल डाई इमीडिएटली, राइट. बट इफ़ वी टॉक अबाउट द मेल्स, फीमेल कैरियर हो सकती है क्योंकि उसका जो काउंटर पार्ट है दूसरा क्रोमोसोम वो कैसा है, नॉर्मल है. बट मेल्स में एक ही एक्स क्रोमोसोम है तो दे में शो द डिसीज, राइट, क्योंकि उसका काउंटर पार्ट वाई है तो हो गया. Now this is sex-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny has been widely studied. So basically females are the carriers and they transmit, they are unaffected but they transmit the diseases to their son. In this disease what is the effect in this, जैसे वहाँ पे क्या हुआ, रेड या ग्रीन कोन में डिफेक्ट आई, यहाँ पे वी हैव स्टडीड द ब्लड क्लॉटिंग मैकेनिज्म. देर आर 13 क्लॉटिंग फैक्टर्स. सो वन ऑफ द क्लॉटिंग फैक्टर इट गेट्स अफेक्टेड, वो बनता नहीं है, वो बनता नहीं है यानी पूरी क्लॉटिंग की जो कास्केड है, ये चारों जो 13 प्रोटीन साथ में काम करते हैं वो कर नहीं सकता है, जिसकी वजह से एक सिंपल भी कट इंडिविजुअल में होगा, देन इट विल रिजल्ट इन द नॉन स्टॉप ब्लीडिंग. So it is a disease in which a single protein that is a part of cascade of proteins, total 13 proteins involved in the clotting of the blood is affected. Due to this, an affected individual single cut will result in a non-stop bleeding. The heterozygous female, that is the carriers, may transmit the disease to their sons. The possibility of a female becoming hemophilic is extremely rare and हुआ तो वो कैसा होगा, very dangerous, because in the recessive condition यानी दोनों genes recessive है तो क्या chances है, ये lethal genes है, an individual dies immediately because mother of such female has to be at least carrier and the father should be hemophilic, which is unviable in the later stage of the. ये कब possible होगा कि फादर हीमोफिलिक है, मदर कैरियर है, तो चांसेस हो सकते हैं कि फीमेल हीमोफिलिक है, लेकिन ये बहुत बहुत रेयर कंडीशन है. द फैमिली पेडिग्री ऑफ क्वीन विक्टोरिया शोज अ नंबर ऑफ हीमोफिलिक डिसेंडेंट्स एज शी वाज द कैरियर ऑफ अ डिजीज. क्वीन विक्टोरिया शी वाज द कैरियर ऑफ हीमोफिलिया एंड उसका फैमिली ट्री जब स्टडी किया गया तो बहुत सारे मेंबर्स ऐसे थे कि दे वर affected with the hemophilia. Now see the inheritance pattern. Mother is a carrier, father is unaffected. The capital H indicates the normal one. ये वाला जो है वो normal है and ये कैसा है, hemophilic है. Same way the father is unaffected, that is normal, and the Y gene. So here you can say there will be unaffected daughter. This is a carrier daughter, this is unaffected son, and this is a hemophilic son. So there are 50% chances that if a carrier mother and the father is unaffected, 50% chances are there that the son may be hemophilic. Now another disease is cystic fibrosis. So what is cystic fibrosis? A recessive mutant allele located on the chromosome number 7, that is autosome. So basically cystic fibrosis, it is a autosomal recessive trait. अगर autosomal recessive trait है, it means what? Both males and females will be affected because the genes are located on the autosomes, right? नाउ इट इज़ अ रेसेसिव ट्रेट. सो अगर नॉर्मल 7 एक एक्स क्रोमोसोम में म्यूटेशन नहीं हुआ है और दूसरे में हुआ है, तो वी नो दैट नॉर्मल इज डोमिनेंट ओवर द अफेक्टेड वन, राइट, एंड अफेक्टेड वन इज द रिसेसिव. सो दोनों अगर रिसेसिव हो गए तो ही ये डिजीज़ होगा, अदरवाइज ये डिजीज़ नहीं होगा. द यूनिक now what is the defect caused in this disease? The unique glycoprotein is produced by a gene which leads to the formation of the mucus of abnormally high viscosity. So यहाँ पे एक प्रोटीन बनता है, ग्लाइकोप्रोटीन बनता है, एंड ये ग्लाइकोप्रोटीन बहुत म्यूकस का फॉर्मेशन करता है जिसकी विस्कोसिटी बहुत ज़्यादा है. The mucus interferes with the functioning of the exocrine glands like sweat glands, liver, pancreas as well as lungs. There is accumulation of a mucus in the long tubules which makes it susceptible to the infections leading to bronchitis. So these are some of the effects of the cystic fibrosis. Now next disease is the sickle cell anemia. Now sickle cell anemia is again a autosomal recessive trait. यानी जीन्स कहाँ पे लोकेटेड है, ऑटोसोम्स पे, यानी बोथ मेल्स एंड फीमेल्स विल बी अफेक्टेड. रिसेसिव है यानी नॉर्मल अगर एक डोमिनेंट है और एक रिसेसिव है तो डोमिनेंट इज गोइंग टू शो द कैरेक्टर. So normal blood is dominant over the sickle cell 1, or you can say normal gene is dominant over the sickle cell gene. It is an autosomal linked recessive trait which is transmitted from parents to offspring when both the partners are carrier for the gene or are heterozygous. So कब ये disease हो सकता है, अगर both male and female partners carrier है and देर आर चांसेस के उनकी जो प्रोजेनी हो वो क्या हो सकती है, दे में बी सिकल सेल. नाउ ये डिजीज़ को एक सिंगल अलील कंट्रोल करता है, HBA ये नॉर्मल है एंड HBS इज़ द जीन रिस्पॉन्सिबल फॉर कॉजिंग द सिकल सेल. नाउ ये 3 जीनोटाइप पॉसिबल है. सो कौन से पॉसिबल है, एक तो HBA ये तो नॉर्मल है, दूसरा है HBA HBS. सो यहाँ पे ए वाला डोमिनेंट है एस पे, यानी नॉर्मल इज डोमिनेंट ओवर द सिकल सेल, एंड तीसरा कंडीशन है HBS. सो बेसिकली जिनका जीनोटाइप HBS है दे विल शो दिस डिसीज. दैट इज हेट्रोजाइगस, दैट इज HBS इंडिविजुअल्स अपीयर apparently unaffected but they are the carrier of the disease as there is 50% probability of transmission of a mutant gene to the progeny thus exhibiting the sickle cell trait. यानी अगर दोनों पार्टनर्स कैसे है, कैरियर्स है, then there is a 50% चांस कि उनकी जो प्रोजेनी हो वो क्या हो, they will be affected by the disease, right? Now ये डिफेक्ट होती कैसे है? So basically in the beta globin chain we know that hemoglobin is made up of 2 alpha chain and 2 beta chain. 2 beta globin chain and 2 alpha globin chain. Globin basically is a protein. Proteins को बनाता कौन है, अमाइनो एसिड. सो बेसिकली बीटा ग्लोबिन चेन में 6th पोजीशन पे ग्लूटामिक एसिड होता है एंड दिस ग्लूटामिक एसिड इज रिप्लेस्ड बाय व्हाट, इट इज रिप्लेस्ड बाय वेलिन. So basically substitution of amino acid in the globin it results due to single base substitution. देखो यहाँ पे एक बेस का ही substitution हुआ है. नॉर्मली GAG होता है, यहाँ पे एक इससे रिप्लेस हो गया है U से, एंड व्हाट इज़ द डिफेक्ट, एक अमीनो एसिड पूरा बदल जाता है. सो ये जो अभी हीमोग्लोबिन बना वो कैसा होगा, म्यूटेंट होगा, नॉर्मल से डिफरेंट होगा. ये हीमोग्लोबिन क्या करता है, इट अंडरगोज पॉलीमराइजेशन अंडर लो ऑक्सीजन टेंशन causing the shape of the RBC from biconcave to elongated sickle. Now we know that RBCs का shape कैसा है, biconcave. Now due to the defect in the hemoglobin molecule and उसका polymerization हो रहा है तो RBCs कैसे बन रहे हैं, सिकल शेप. ये कौन सा शेप हो गया, सिकल शेप. Now ये सिकल शेप की वजह से उनका जो साइज है वो इंक्रीज हो गया, they have become elongated. एंड ये इलोंगेटेड आरबीसीज को ब्लड वेसल से पास करना इट इज़ डिफ़िकल्ट फॉर देम एंड ड्यू टू दैट दिस आरबीसीज गेट रप्चर्ड रिजल्टिंग इनटू द अनीमिया. सो बेसिकली द सब्स्टिट्यूशन of the amino acid results due to the single base substitution. एक छोटा सा बेस ही क्या हो रहा है, रिप्लेस हो रहा है, जिसकी वजह से कोडोन ही पूरा चेंज हो रहा है. नॉर्मल कंडीशन में क्या होता है, GAG, लेकिन म्यूटेशन की वजह से क्या हो गया, GUG. See the inheritance pattern. See, these both are the, ये भी carrier है, HBS, and the other partner is also the carrier. Now see the condition. ये दोनों हुआ तो ये normal individual है, ये दोनों हुआ that is HBS, that is carrier. Again this with this, that is HBS, we know that A is dominant over S, so carrier. But see the chances that if both the recessive genes are found, then the person will be a sickle cell person, right? Now see how this disease occur. नॉर्मल पोजीशन पे क्या है, GAG है, राइट. ये mRNA एंड ये GAG सिक्स्थ पोजीशन में कौन सा अमाइनो एसिड रिप्रेजेंट करता है, ग्लूटामिक. बट ये जीन की वजह से म्यूटेशन क्या हुआ कि यहाँ पे क्या हो गया, ये U हो गया, एंड ड्यू टू दैट इंस्टेड ऑफ द ग्लूटामिक एसिड क्या आ गया, वेलिन. So that is the mutation occurring in the genes. Now next disease is phenylketonuria. Again it is an inborn error of metabolism कि जहाँ पे autosomal recessive trait है. Autosomal recessive यानी normal is dominant over the affected one, and autosomes पे genes है यानी both males and females may be affected. Now यहाँ पे क्या होता है कि there is an chemical called as phenylalanine, and ये phenylalanine किस में convert होता है, tyrosine, in the presence of the enzyme that is called as the phenylalanine hydroxylase. So in a normal individual, the phenylalanine, in the presence of phenylalanine hydroxylase enzyme, it gets converted into tyrosine. But in a person suffering from phenylketonuria, this enzyme is lacking. As enzyme is lacking, phenylalanine is not converted into tyrosine. As a result, the amount of the phenylalanine goes on increasing and it gets accumulated in the body. And as it gets accumulated, it is converted into phenylpyruvic acid and some of its derivatives. Now, accumulation of these all compounds in the brain, it results into mental retardation. It also causes the light pigmentation of the skin and hair, and it is also excreted through the urine because it is poorly absorbed by the kidney. So that is regarding the phenylketonuria, which is an autosomal recessive trait. Now the next disorder is thalassemia. Again it is an autosomal recessive disorder, yani normal is dominant over the recessive one. And the genes are located on the autosomes, so both males and females will be affected. It is transmitted from parents to offspring when both the partners are unaffected carriers of the gene. The defect could be due to the either mutation or deletion, which ultimately results in the reduced rate of synthesis of one of the globin chains that make up the hemoglobin. Now students, what is thalassemia? We know that a hemoglobin is made up of 4 globin chains, 2 alpha globin chains and 2 beta globin chains. Now यहाँ पे क्या डिफेक्ट होती है कि एनी ऑफ द चेन में म्यूटेशन होता है, जिसकी वजह से ग्लोबिन चेन का सिंथेसिस कम हो जाता है. The mutation resulting in the excessive destructions of the RBCs due to the formation of the abnormal hemoglobin molecules. हीमोग्लोबिन एबनॉर्मली बनते हैं, उसकी क्वांटिटी भी कम होती है, जिसकी वजह से RBCs रप्चर होते हैं and it results into the anemia. Now we know normally hemoglobin is composed of 4 polypeptide chains, 2 alpha and 2 beta globin chains. The thalassemia patients have defects either in the alpha or beta chain, causing the production of abnormal hemoglobin molecules, which results in the anemia. Now, based on which chain is affected, there are 2 types of the thalassemia. One is alpha thalassemia and another one is the beta thalassemia. अगर alpha globin chain की formation में defect आई, then it is alpha thalassemia. बीटा चेन के फॉर्मेशन में अफेक्ट आई, देन इट इज़ कॉल्ड एज बीटा थैलेसेमिया. So thalassemia is classified into alpha and beta based on which chain of the hemoglobin molecule is affected. Now, it is controlled by the two closely linked genes. Basically, we are talking about the alpha thalassemia. So, alpha thalassemia, it is controlled by the two genes which are located. Names of the genes are HbA1 and HbA2. And where are these genes located? They are located on chromosome 16 of each of the parents. So basically, there are two linked genes HbA1 and HbA2 on chromosome number 16. Now अगर mutation हुआ, दो genes are present in each parent. So offspring में total कितनी होगी? 4. So mutation or deletion in one or more of the 4 alpha gene causes the alpha thalassemia. सो टोटल कितनी जीन्स होगी? HbA1 भी 2 है, HbA2 भी 2 है. सो टोटल कितनी जीन्स है? 4. ये 4 में से एक में भी म्यूटेशन आया या ज़्यादा में म्यूटेशन आया तो अल्फा थैलेसेमिया होगा. यानी द मोर द जीन्स अफेक्टेड, लेस अल्फा ग्लोबिन चेन्स आर प्रोड्यूस्ड. Basic and what is in beta thalassemia? The production of the beta globin chain is affected. Here it is which we are talking about. We are talking about the alpha thalassemia, and here we are talking about the beta thalassemia, जहाँ पे beta globin chain का production affect होता है. Now alpha globin chain के synthesis को दो genes control करती है, HbA1 and HbA2. जबकि ग्लोबिन चेन बीटा थैलेसेमिया, it is controlled by a single gene HBB which is present on the chromosome number 11 of each parent. यानी मेल गैमेट कितने हैं जीन्स एक पेरेंट की? सो बेसिकली सिंगल जीन HbA1 which is present on the chromosome HBB which is present on the chromosome number 11. So ऑफ कोर्स मदर की एक जीन आएगी, फादर की एक जीन आएगी, तो टोटल कितने जीन्स होगी इंडिविजुअल में? ऑफस्प्रिंग में कितनी जीन होगी? 2. अगर एक में या दोनों में डिफेक्ट आई देन द डिजीज विल टेक प्लेस. सी स्टूडेंट्स, अगेन आई एम टेलिंग यू, basically there are two globin chains, two types, alpha chains and beta chains, right? Now the formation of the alpha chain is controlled by the two genes. One is HbA1 and HbA2. So in two genes है, तो genes occur in pair. So total कितनी genes है? 4 genes. अगर इंडिविजुअल में एक जीन में डिफेक्ट आया या मोर देन वन जीन में अफेक्ट आया देन अल्फा थैलेसेमिया होगा. एंड बीटा थैलेसेमिया को कितने जीन कंट्रोल करते हैं? एक, HBB. वो कौन से क्रोमोसोम के ऊपर लोकेटेड है? 11th क्रोमोसोम. नाउ स्टूडेंट्स, जीन अकर इन पेयर. सो इंडिविजुअल, पर्टिकुलर इंडिविजुअल में कितनी वो जीन होगी? बीटा ग्लोबिन चेन के लिए 2 होगी. सो अगर एक में डिफेक्ट आया या दोनों में डिफेक्ट आया देन बीटा थैलेसेमिया होगा. Beta thalassemia, it is the most common type of the thalassemia and it is also called as Cooley's anemia. Basically, in this disorder, the alpha chain production increases and damages the membranes of the RBCs. Cooley's anemia is the beta thalassemia, जिसकी वजह से अल्फा चेन का सिंथेसिस बहुत ज़्यादा हो जाता है एंड वो RBCs के मेम्ब्रेन को डैमेज करता है. Now, what is the difference between thalassemia and sickle cell anemia? Thalassemia वो डिजीज़ है जहाँ पे क्वांटिटेटिव डैमेज है, दैट इज ग्लोबिन मॉलिक्यूल्स कम सिंथेसिस होते हैं. सो इट इज़ अ क्वांटिटेटिव डिफेक्ट. जबकि सिकल सेल एनीमिया वो कैसी है? हीमोग्लोबिन की चेन्स बन रही है बट क्वालिटी प्रॉपर नहीं है, राइट? सो इट इज़ अ क्वालिटेटिव, ये अनइफेक्टिव और यू कैन से इनकरेक्ट हीमोग्लोबिन सिंथेसिस होता है. जबकि थैलेसेमिया में कम नंबर में सिंथेसिस होता है हीमोग्लोबिन का. So thalassemia, it differs from sickle cell anemia in that the former that is we are talking about the thalassemia, it is a quantitative problem of synthesizing too few globin molecules, while the latter that is we are talking about the sickle cell is a qualitative problem of synthesizing an incorrectly functioning globin. So that is all regarding the Mendelian disorders. Now we will deal with the chromosomal disorders. So they are basically caused due to the absence or excess or abnormal arrangement of one or more chromosomes. नाउ ये कब हो सकता है? फेलियर ऑफ द सेग्रीगेशन ऑफ द chromatids during the cell division results in the gain or loss of the chromosome called as the aneuploidy. We know that cell division के टाइम पे chromatids का segregation होता है, separation होता है. So ये अगर नहीं हुआ then it results in the gain or loss of the chromosomes. एग्जाम्पल डाउन सिंड्रोम के जहाँ पे देर इज़ एन एक्स्ट्रा कॉपी ऑफ द क्रोमोसोम नंबर 21. क्रोमोसोम नंबर 21 कितने होते हैं? 2 होते हैं, बट यहाँ पे एक्स्ट्रा हो जाता है. सो आई टोल्ड यू व्हाट इज़ दैट कॉल्ड एज ट्राइसोमी, राइट. सिमिलरली टर्नर सिंड्रोम में क्या होता है? ड्यू टू द लॉस ऑफ एक्स क्रोमोसोम इन द ह्यूमन फीमेल्स. कितने एक्स क्रोमोसोम होते हैं फीमेल में? 2. So instead of 2 there is only one sex, that is monosomy of the sex chromosome. So failure of cytokinesis after the telophase of the cell division. We have studied the types of cell division, mitosis, meiosis. So cell division के telophase के end में बाद में क्या होता है? cytokinesis होता है. So cytokinesis अगर नहीं हुआ then it results in the increase of the whole set of the chromosome and this phenomenon is called as polyploidy. ओके चलो, प्रॉपर सेल डिवीजन हो गया, क्रोमोसोम का डिस्ट्रीब्यूशन प्रॉपरली हो गया. ओके एंड में वो क्रोमोसोम्स 2 पोल पे आ जाते हैं, राइट, टीलोफेज पे. बाद में टीलोफेज के एंड के बाद क्या होता है? साइटोकाइनेसिस के, जहाँ पे सेल मेम्ब्रेन का फॉर्मेशन होके 2 सेल्स बन जाते हैं. बट अगर साइटोकाइनेसिस नहीं हुआ तो क्या हो जाएगा कि एक ही सेल में 2 सेट्स ऑफ क्रोमोसोम हो जाएंगे. सो व्हाट इज़ दैट कंडीशन कॉल्ड एज पॉलीप्लॉइडी. And it is often seen in the plants also. Now human being से बात करते हैं. We have 46 chromosomes, that is 23 pairs. Out of this 22 are autosomes and 1 pair is the sex chromosome. Sometimes, very rarely, either an additional copy of the chromosome may be included in an individual or individual may lack one of the pair of the chromosomes. These situations are known as trisomy or monosomy respectively. अगर एक कॉपी ऐड हुई तो ट्राइसोमी, अगर क्रोमोसोम में से एक कम हो गया तो मोनोसोमी. This leads to the very serious consequences in the individual. Some of the consequences are Down syndrome, Turner syndrome, Klinefelter syndrome. We will study them, students. So this is the Down syndrome. So it is a genetic disorder in the presence of the additional copy of the chromosome number 21. कितने क्रोमोसोम 21 होते हैं? 2, बट यहाँ पे एक एडिशनल है. सो एक एक्स्ट्रा हो गया तो क्या हो गया? ट्राइसोमी. वी हैव स्टडीड दिस इन म्यूटेशन. सो व्हाट इज डाउन सिंड्रोम? इट इज़ अ ट्राइसोमी ऑफ द क्रोमोसोम नंबर 21. Now as it was first described in Langdon Down, it is called as the Down syndrome. Now what is the symptoms of this disease? So individual is short statured, उसकी हाइट कम होती है, स्मॉल राउंड हेड होता है, फरोड टंग होती है एंड पार्शियली ओपन माउथ. palm is broad with the palm crease. The physical, the psychomotor, that is origination of the movement in the conscious mental activity, ये नहीं होता है. and mental development is retarded. यानी फिजिकल डेवलपमेंट भी नहीं होता है, साइकोमोटर डेवलपमेंट भी नहीं होता है, एंड मेंटली ऑल्सो द पर्सन विल बी रिटार्डेड. So here you can see this is the 21st chromosome where there are how many chromosome? 30 chromosomes, and this diagram it is called as the karyotype. इसको क्या बोलते हैं? karyotype. karyotype के जहाँ पे हम क्रोमोसोम्स का अरेंजमेंट प्रॉपरली देख सकते हैं. सो हियर आप डायग्राम में देख सकते हैं कि टंग इज रिंकल, फरोड, ब्रॉड फ्लैट फेस, हाइट कम है, देन हेड इज फ्लैट, लूप्स आर देयर इन द फिंगर्स एंड पाम में क्रीज है. नाउ नेक्स्ट इज द क्लाइनफेल्टर सिंड्रोम. So basically it is due to the presence of an additional copy of X chromosome into a karyotype of, that is the males have the extra X chromosomes. Normally males में कितने X chromosome होते हैं? X, एक ही होता है, but instead of one they have the 2 X chromosome. So basically it is a trisomy of the sex chromosome in the males. Now such an individual has a overall masculine development, वो दिखेगा masculine, but however the feminine development, that is development of the breast, which is called as the gynecomastia, is also expressed, and individuals are sterile. sterile यानी उनमें gametes का formation नहीं होगा, स्पर्म्स का फॉर्मेशन नहीं होगा. सो क्लाइनफेल्टर सिंड्रोम, इट इज़ द ट्राइसोमी ऑफ द एक्स क्रोमोसोम इन द मेल्स. एक एडिशनल एक्स क्रोमोसोम मेल्स में आ जाए. नॉर्मल कितने क्रोमोसोम्स होते हैं? 46. इंस्टेड ऑफ 46 कितने क्रोमोसोम्स हो गए? 47. कौन सा बढ़ा? सो एक एक्स क्रोमोसोम इंक्रीज हुआ. सो इट इज़ अ ट्राइसोमी ऑफ अ सेक्स क्रोमोसोम इन द मेल्स. Another one is the Turner syndrome. It is caused due to the absence of one X chromosome. That is normally again ये disease females की है. So ये disease में क्या होता है? Females के पास total कितने X chromosome होते हैं? 2. Instead of 2 they are having only 1. So it is monosomy of the sex chromosomes. So total chromosomes कितने हो जाएंगे? Instead of 46, 45 हो जाएंगे. Females are sterile as ovaries are rudimentary. Rudimentary ovaries means they are not well developed ovary. Well developed ovaries नहीं है यानी gametes का formation नहीं होगा, eggs का formation नहीं होगा. Besides other दूसरे features क्या है? They lack the secondary sexual characters. Of course ovaries proper नहीं है यानी sex hormones का secretion नहीं होगा. Sex hormones का secretion proper नहीं होगा तो secondary sexual characters नहीं होंगे. So students, here you can see a person. The first one is the Klinefelter syndrome, which is in the males, and this is the Turner syndrome in the female, right? So here you can see the development of the breast is more because it have an additional X chromosome, and here the ovaries are not well developed. In the next diagram it will be more clear to you. सी दिस इज़ अ टर्नर सिंड्रोम X0. यहाँ पे फीमेल्स में एक एक्स क्रोमोसोम कम है. क्लाइनफेल्टर सिंड्रोम XXY जहाँ पे मेल्स में एक एक्स क्रोमोसोम ज़्यादा है. सो द मेन थिंग इज़ हियर यू कैन सी द ओवरीज आर rudimentary. Due to that there won't be the menstruation as well as gametes formation भी नहीं होगी. And here you can see that the breast development is more because of the additional X chromosomes. And testes atrophy, that is उनके tissues damage हो जाते हैं. So this is one of the symptoms of the Klinefelter syndrome. So today students, we have ended this particular chapter. We have studied today pedigree analysis. We have studied the Mendelian as well as the chromosomal disorders. So we end our chapter here. We will begin the next chapter in the next video. Thank you students.